Vitis vinifera is considered one of the most widely cultivated crops of global economic importance and is the only grape variety used to produce quality wines. Vitis vinifera is characterized by high genetic and phenotypic variability, but selection carried out by humans has restricted cultivation to a relatively small quantity of varieties selected for their yield and quality related characteristics, such as berry composition, phenotype, and resistance to stress. Vitis vinifera varieties widely grown worldwide are vulnerable to a variety of phytopathogens that could cause severe crop loss. This not only has a negative impact on the financial status of farmers, human health, and the environment, but may also reduce future disease control to the development of fungicide resistance. In this study, we focused on disease and pest resistance in Vitis vinifera and how these disease and pest resistance influence growth and how they became more resistant. Powdery mildew caused by the fungus Erisiphe necator is an important disease of grapevines in Australia and worldwide. It appears as a characteristic grey-white powdery growth on a green tissue of the grapevine. The cultivated grapevine, Vitis vinifera, has no natural genetic resistance to powdery mildew. This is due to the two organisms evolving on different continents. Vitis vinifera first evolved in Europe while the pathogens came from North America. For this reason, the only method growers must control these pathogens is by using fungicides. There are certain Vitis species that are more resistant to powdery mildew infection than others. For example, the European varieties belonging to Vitis vinifera are not typically resistant, while species native to North America have co-evolved and often carry resistant R genes because of adaptive selection. In grapevines, the effect of powdery mildew ultimately leads to a reduction in yield and loss of berry and wine quality. Phylloxera is a sap-sucking insect native to North of America that feeds on grapevine roots and leaves. The insect is viticulturally known as one of the most deformative and resilient pests and is extremely hard to get rid of. The secondary fungal infections caused from the insect cut off the flow of nutrients and water to the vine completely. This causes complete necrosis of the vine. It is known for the phylloxera epidemic in the 19th century Europe, where 90% of Europe's vines were destroyed. Vitis vinifera is a European grape species that is extremely susceptible to the insect and cannot survive once the eggs have hatched. Phylloxera has a complex 18-stage life cycle where the eggs hatched by the nymph are born into the vine's leaves. This is done parthenogenically. It is for the reason that once the pest has hatched itself into the vine, there is no cure or chemical that can terminate it. Although there are some North American rootstocks that have built up several natural defences against the insect, such as Vitis labrusca. These roots excrete a sap that repels the nymph by clogging its mouth when it tries to latch onto the vine. They also form a protective layer of tissue to cover the vine's wounds. This prevents any secondary infections. Therefore, Vitis vinifera alone is not resistant to phylloxera, but there are certain rootstocks of the species that are. Nematodes are microscopic roundworms that are parasitic pests affecting different agricultural crops, including the grapevine. The parasites are found in the soil, puncture the cell wall of root cells, causing malformations or necrosis. This, in turn, results in a nematode-infested vineyard, displaying reduced yields, vigour, and increased susceptibility to other stresses. Nematodes are a worldwide industry problem, but in Australia, the pest has been estimated to cost $14 million per year. Meloidigine javanica has been identified as the most predominant nematode in Australia. Once introduced to a vineyard, a nematode infestation is permanent. Some strategies, such as deep tillage, can reduce nematodes' impacts, but the main solution viticulturists have employed are resistant rootstocks. Some varieties of Vitis vinifera alone are resistant to nematodes, for example, the sultana vine, but many are susceptible. Studies have shown that some cross rootstocks of Vitis vinifera provide greater resistance, for example, the common Ramsay cross. Vitis vinifera typically has a low resistance to common pathogens and pests such as powdery mildew, phylloxera, and nematodes, given they evolved on different continents. Due to this low resistance, it is quite common for viticulturists and vineyard managers to use rootstocks to combat the issue, wherein one vine species is grafted to another to create a hybrid with desirable characteristics taken from each parent. The main parental rootstock species all originate from North America, and each can offer different advantages and disadvantages in the combat of pest and disease. 4 main rootstock parental species that are used as hybrids and grafted to fruit-producing Vitis vinifera are Vitis berlanderi, Vitis rupestris, Vitis champini, and Vitis riparia, each offering different advantageous characteristics. Rootstocks that offer high resistance to phylloxera are hybrids of Vitis berlanderi and Vitis rupestris, such as 1103 Paulson, 140 Ruggieri, 99 Richter, and 110 Richter. Hybrids of Vitis berlanderi and Vitis riparia typically show good resistance to phylloxera and nematode rootstocks, such as SO4, 5C Telki, and 5BB Cobra. Finally, hybrids between V. champagnei and V. riparia offer exceptionally high resistance to both phylloxera and nematodes. These include K5132, K5140, and straight V. champagnei rootstock Ramsay, which has become increasingly popular through Australian vineyards due to its favourable resistance against pests. It is important to consider in the selection of rootstocks that, whilst they may be resistant to pest and disease, they may have shortcomings against other factors relating to soil, climate, and planting requirements. These factors all need to be balanced to find the ideal rootstock for the growing conditions. There is still a lot of research to be performed to understand the best way to manage disease and pests in Vitis vinifera. In saying this, Vitis vinifera resistance has developed to be able to identify the pest and disease in which is damaging the grapevine and therefore know how to treat it. Grapevine growers are faced with the challenges of permanent diseases such as phylloxera, nematodes, and powdery mildew. Over time, the Vitis vinifera has adapted to these harmful pests, with different varieties of the Vitis vinifera now being resistant due to the implication of rootstocks, whereby one species of Vitis is grafted to another, utilising those favourable qualities, increasing the resistance of the species.